Hello and welcome to chapter 14, which is going to be the overview of sort of how genes are expressed to make proteins and proteins that then express traits. So this chapter should do a good job of covering how we go from DNA to brown eyes or curly hair. So what do we need to know? In terms of the overview of the flow of genetic information, we know that genes are coded by specific sequences of nucleotides in DNA. We have 4 specific nucleotides, A, G, T, and C. And we know that proteins are sort of the physical expression of traits that allow humans to have unique characteristics within cells and tissues and organisms. A good example would be the protein in cystic fibrosis that doesn't allow chloride ions to leave, which then has huge implications on the phenotypic expression of a physical human body. Now, when we refer to gene expression, we are referring to the process in which DNA is going to be directly communicating and inducing and producing protein synthesis. And there's 2 key stages: transcription and translation. And we can really think about protein production and gene expression and different patterns of inheritance when we look at something like albinism, the lack of pigment production in cells versus normal individuals. So all of this science is based off of a lot of discoveries in the past 200 years, and one of those discoveries was Beadle and Tatum working with bread molds where they disabled genes and looked for phenotypic changes within those types of molds. And they studied the haploid bread mold because it would be easier for them to detect either recessive mutations or differences in alleles. And what they did was they studied mutations that altered the ability of the fungus to grow on a minimal medium. When we talk about a minimal medium, we're talking about a growth media that doesn't really have a lot of available amino acids or carbohydrates. So it's basically the bare minimal needed for molds to survive. And what they did was they would collect these neurospora cells, which are molds that grow in bread. I'm sure you've all seen mold growing on old food before. They would then subject them to X-rays to induce random mutations. Each of the surviving cells would form a colony of genetically identical cells because they're haploid and they do asexual reproduction. And then they would put those growths in different media to see whether or not they would survive in different types of media, whether it was the minimal media or the media with additional nutrients. And this sort of helped them realize that probably that there were specific genes coding for specific proteins that allowed these fungi to basically create enzymes that would allow them to digest different types of nutrients. And this sort of helped develop the idea of the one gene, one protein hypothesis. Now, in terms of understanding the gene expression of protein, we really have to think about DNA, which is going to be coded into RNA, and then that RNA is going to be used by ribosomes in the ER to make our proteins. We have 2 different types of this process in terms of transcription and translation. Because bacterial cells lack a nucleus, transcription and translation are going to just take place in the cytoplasm. And there's going to be mRNA being created from transcription, which will be directly fed into ribosomes, which are free-floating because bacterial cells lack the endoplasmic reticulum, and that will make a polypeptide. That's pretty straightforward. In eukaryotic cells, transcription will occur within the nucleus. We will make a pre-mRNA transcript. There will be some RNA processing, much like what the Golgi apparatus does to proteins, and that mRNA will then be exited out of the nuclear pore to be fed into either a free or bound ribosome. Remember, free ribosomes are going to make proteins that stay within the cytoplasm, and bound proteins are going to be in the ER that make the polypeptides into a transport vesicle because they're going to be exocytosisly leaving the cells. So good examples would be hormones or signal molecules. So here is the basic process of how this is going to work. We are going to be reading our template strand, and that template strand might be 3 to 5 or 5 to 3. Whatever template strand is going to be used for the DNA, we are going to have the opposite anti-parallel transcript of mRNA. So if this is our template strand, 3 to 5. Notice this will be 5 to 3. The only thing that's new in this process is that we're going to be making a single strand of nucleotides, not a double strand, and that we are going to be replacing the nucleotide thymine with the nucleotide uracil. G's still goes with C's, A's will still code for U's, etc., etc. And then we do translation, where we read every 3 nucleotides as a codon, which creates an amino acid. Now, what we need to know about this process is that there are only 20 amino acids within the human body, but there's 64 different possibilities in terms of codon organization, because there is 4 possibilities in the first, second, and third nucleotides. 4 to the third power is 64. Generally, there will be a few codons that we all need to know. OG usually codes for methylene, or the start of an amino acid polypeptide chain, and a few codons are stop codons, which induce this cessation of creating a polypeptide. So we first start off with just normal transcription, and transcription is going to be DNA-directed synthesis of RNA. And we usually start with a promoter. A promoter could be one or more proteins that attach to the double-stranded segment of DNA. And this is generally going to be a region of the DNA that does not code for the protein, but contains genetic information that will help initiate the beginning of our transcription. Once the promoter binds to the DNA and we unwind it, we are going to have RNA polymerase bind to the coding strand of the DNA, and it is going to start making an RNA transcript. And the RNA transcript is always going to be made in a 5' to 3' direction, which means generally the coding template is going to be a 3 to 5' in terms of DNA. As the promoter region moves, as the promoter moves down, we are going to have the DNA become rewound. So the direction of transcription will always be downstream of the 3 to 5'. When we get to a region that generally codes for the termination of it, it might be a region of DNA that is highly methylated or highly condensed. The promoter region will leave, RNA polymerase will end, and we'll have a completed RNA transcript. And promoters really are these proteins that really signal the transcriptional start point, and they usually extend several dozen nucleotide pairs upstream of this starter point. And transcription factors sort of help mediate the binding of RNA polymerase. You can think of them as the helping friends or the wingman of RNA polymerase to get transcription started. And many transcription factors are proteins that are activated or go into the nucleus because of cell signaling. They're often secondary messengers or the final response in a signal cascade. So the completed assembly of transcription factors and RNA polymerase are going to bind to that promoter, and this is called the transcription initiation complex. A promoter region is often called the TATA box, and it's crucial in terms of the initiation complex in eukaryotes. It's called a TATA box because generally the region is rich in thymines and adenines, which only have 2 hydrogen bonds per base pair. And that means it's a slightly easier segment of DNA to unwind than guanine and cytosine, which have 3 hydrogen bonds. You can think of hydrogen bonds as Velcro, so the more bonds you have in a region, the harder it is to separate. Now we can talk about eukaryotic transcription. And this is going to be the transcription factors sort of binding the DNA. Usually those factors are going to sort of unwind or relax the DNA, which will allow RNA polymerase to come into the opened region, and it will start undergoing that transcription. The promoter region, notice, is always going to be upstream of the template strand and the coding segment of DNA. And this is sort of what we see at a very micro level, where we have the RNA polymerase adding in nucleotides in a 5' to 3' direction. And then the template strand is going to be re-engaging with the non-template side of DNA as soon as this RNA polymerase moves. Now, in terms of eukaryotic modifications, mRNA molecules are modified in a variety of ways. The 5' end, which is going to again be the sort of initial end of the DNA, is usually going to get a modified G nucleotide cap. And this generally is a modified guanine that assists in protecting that side of it. And then the 3' end usually gets a poly A tail, where we have a duplicitous, repetitive segment of adenines that really don't do anything for coding a protein, but again, are really important for protecting the mRNA from enzymes. So these two modifications protect, help facilitate export, and also help the ribosome attach to the 5' end so it knows the direction of coding. And this is sort of what we see when we look at an mRNA transcript within a eukaryotic. We have this 5' end guanine cap. We have our start codon. We have a protein coding segment. We have a stop codon, and then we have a very long poly A tail, which sometimes can be 50 to 250 adenine nucleotides in a row, which is pretty miraculous. Now, most eukaryotic mRNAs have long non-coding stretches of nucleotides that lie between coding regions. This could be from mutations or random coding errors during eukaryotic transcription. Some of it could be from viral insertions of DNA. And these non-coding regions we don't want when we actually get to the finished product of a protein. These would be the long superfluous sentences you put in your English paper that sometimes get X'd out, and you hear your teacher say, this is just too much information. We call these non-coding regions introns. The regions that we want to stay in our mRNA transcript are called exons, and these usually contain nucleotide sequences that directly code for amino acid sequences. RNA splicing occurs where we remove the introns and we join the exons together to create a continuous coding sequence. So generally we will have introns at certain points of the nucleotides, and they will be spliced out, and then we will use DNA ligase to reattach these introns together to make a coding segment. Notice we don't remove the poly A tail or the 5' guanine cap. And spliceosomes are these protein RNA complexes that use small strands of RNA which code complementary to the introns to help remove the exons. So you can always think of spliceosomes as splicing out the information, and we use complementary base pairs to achieve this splicing. And ribosomes also do this. Ribosomes are RNA molecules that function as enzymes, and they can occur without proteins to also get rid of the introns. And then we get to translation, and translation is going to be where a ribosome uses the codon sections of a mRNA transcript to code for a polypeptide. Now, we need to talk about a few key actors in this process. One is sort of the anticodon and the tRNA transfer molecule. So when we look at this tRNA molecule, we will see that there's always going to be this sort of loop at the bottom, which codes for the opposite 3 nucleotides. So AAG would probably be coding for a UUC. So these anticodon codes for the codon that we find at the nucleotide. We also have a 3' end here, which is called the amino acid attachment site. This is going to be a repetitive nucleotide region that attaches to an amino acid during and before protein production. So a ribosome has 3 binding sites for these tRNA molecules, which means at any given point we might see 2 or 3 of these tRNA molecules with their anticodons within a ribosome. I always think about the acronym APE. So we have P, which is going to be the tRNA that's carrying the growing site. This is always in the middle. We have the A site, which is to the right of the P protein, and this is going to be where we have the next amino acid that's going to be added to the chain. And then finally we have the E site, which is the exit site. This is where the tRNA that no longer has an amino acid leaves the ribosome. So generally during translation, we first have our start codon, which is AUG, which will code for an anticodon of UAC, 5' to 5'. And that is almost always going to code for the amino acid methylene. And this is going to be occurring in the P site of our ribosome. And here's our large and small subunit. We're going to use some GTP to have this attachment occur, because GTP is a form of energy, which means it can make proteins change shape and initiate functionality. The GTP is then going to be used to attach another tRNA molecule based on the anticodon and codon regions. And then the peptide bond between these two molecules, remember it's a covalent bond, so we're going to be sharing electrons and doing dehydration synthesis, removing a water molecule, will form a polypeptide, and that continues. And every time we have the A shift to the P, and the P shift to the E, and then the E tRNA exits. Termination occurs when we reach a stop codon in the mRNA, and that is often going to first start at the A site, and then the A site accepts that protein, which will then cause a release factor to come, and that release factor causes the addition of a water molecule instead of an amino acid, and this water substitution releases the polypeptide from the ribosome, and the assembly comes apart. So we'll have a release factor come instead of a tRNA. That tRNA will cause, or this release factor will cause the tRNA to move to the E site. The polypeptide will be free, and then the ribosome disassembles using some more GTP. Now, there are some modifications and post-translation modifications that occur. So during synthesis, a polypeptide chain just starts to spontaneously coil and fold based on hydrogen bonds to make the alpha helix, and then R groups to make the tertiary structure. We often have post-translational modifications that need to occur. We have free and bound ribosomes, and free ribosomes are going to be the ribosomes that synthesize proteins that function within the cell, and bound ribosomes are going to be made in the endoplasmic reticulum, and they are often secreted from the cell. A signal recognition particle is going to bind to any signal peptide that is going to sort of help bring about a ribosome knowing that it needs to make a protein in the ER. So generally what happens is if we know that we are producing a ribosome, or not a ribosome is producing a protein that we have deemed necessary to go into the cell membrane or leave the cell, it's going to have amino acids at the beginning of its polypeptide region that can bump into a unique SERP, and that recognition will then lead the ribosome to float to the membrane of the ER. And this is going to be how we then have a protein produced into a transport vesicle. And the same thing will happen where eventually we'll reach a stop codon at the A site, which will lead to the protein being completed and the translation assembly line being disassembled. Now, humans and other eukaryotic organisms are obviously complex creatures with trillions of cells and millions of different proteins that we produce, and we also live in a very fluctuating environment that needs to have homeostasis occur every single second of every minute of every day, and we need to maintain that homeostasis pretty rapidly. So we often find that within a certain gene or protein complex, that we can often have what's known as a polyribosome, which means that we can have one mRNA transcript flow through multiple ribosomes to create a protein assembly line, where we would have the 3' end of the mRNA flowing through, or the 5' mRNA flowing through all of these proteins, which will lead to many proteins being produced. So the 5' started in this ribosome, and then this ribosome, and this ribosome, and this ribosome, which allows us to produce the same protein many times over from just one transcript. And this is a really easy way for us to have really fast and responsive changes to whether it's cell signaling or protein production or maintaining homeostasis. And this is the full overview of the process, where we have our DNA region that codes for a protein. We make an RNA transcript. We have exons remain, introns are cut out. The poly, A tail, and the G cap help maintain the mRNA's sort of structure and integrity, because the cytoplasm is a very nasty place for a very susceptible RNA transcription. The RNA will leave the nuclear envelope to go out to the cytoplasm, either bind to a free or bound ribosome, and have tRNA bring in different amino acids. Now, there are a few mutations that can really hamper and damper and influence whether or not a protein maintains its structure and its function. And mutations are just changes in the genetic material of any cell or virus based on the code of nucleotides. And one type of mutation that can occur are point mutations. And point mutations are just chemical changes in one or a few nucleotide pairs of any gene. And the change of a single nucleotide in any DNA template can really influence the production of an abnormal or different protein. So when we look at sickle cell anemia, this is a great example of a point mutation where in a codon, a T is substituted for an A, and that's going to then correspond to a change in the codon. So we're going to go from GAG to GUG, and the GAG codon codes for a hydrophilic glutamine acid, while the GUG codon codes for a hydrophobic valine. And that has some really important implications for why the red blood cells in sickle cell anemia individuals sort of clump up together because of that hydrophobic interaction. We can also have the introduction of a stop codon, or I apologize. We can have the introduction of what's called a silent mutation, where the genetic code results in the same protein despite a change in the codon and the nucleotide in the DNA. And this generally occurs in the third nucleotide, which we often call the wobble nucleotide, because the codon GGU and GGC both code for a glutamine. However, if we have a change in the first or second nucleotide, that almost always results in a missense mutation, where we have a completely different codon, AGC versus GGC, so we make a glutamine instead of a serine. We can also have what's called a nonsense mutation, where we have a mutation pretty early on in the coding strand and transcript, and that usually causes a stop codon to be introduced prematurely, so we don't make a full-fledged protein. And then we can also have a frame shift mutation, where we actually accidentally have an inserted nucleotide, which sort of shifts all of the codons upstream of it one space over. And this often results in a completely new protein being produced, because we are having all of the amino acids moved over one, or all of the nucleotides moved over one. And then we can also have a frameshift mutation from a deletion where we delete a mutation and suddenly we have new amino acids because we deleted one of many nucleotides in the strand. And we can also have 3 nucleotide pairs where there's no frameshift but one amino acid is missing, so we take out 3 nucleotides. And again, these are all mutations that occur during the S phase of mitosis, where we duplicate DNA and sometimes cells just make mistakes. It's usually 1 in a billion when it comes to the rate of mutations per nucleotide, but they do exist. So we can have frameshift mutations, we can have nonsense mutations, we can have missense and silent mutations. I hope this video has been helpful for chapter 14. I know it's a lot. You guys are going to do great, and I will see you soon.